Hello guys, welcome to Big Book. Today we are going to learn about the cell cycle and the regulation of the cell cycle in eukaryotes. First we will have a brief discussion on the cell cycle, that is increase in cell mass, that is during the cell division, that is the mitotic cell division, or it can be in the meiotic cell division also. There will be the increase in the cell mass, that is the overall cell size is going to increase, and also the whatever the content like cytosol and the other components are going to take their perspective kind of the whatever the molecular mechanisms has to be happened, that they are going to make it changes. Because of that, this size increase in the size of the cell is going to happen. So further followed by that, the main activity is going to be the semi-conservative replication of the genetic DNA. That is, the DNA is going to replicate on its own, where the semi-conservative, that is the mirror copy as per the 5 prime to 3 prime and 3 prime to 5 prime is going to happen. And also after that, the condensation of the DNA is going to take place in the form of the chromosomal sister chromatids, where these are going to form the equal complement, and this equational division of the chromosome is going to push the genetic material to progeny cells. Here further in the next slide, it is initially in the mitotic division, initially a diploid cell undergoes a period of growth and increases in cell mass. Here they are using the diploid cell, that is the 2n number of the organisms, referring to the 2n number of the organisms. Cell undergoes a period of growth and increases in cell mass. So the cell mass is going to increase. During this stage, it will be around 10 hours and so. And this stage is also called as the G1 phase cell cycle. G1 stage is dedicated to cell growth and chemical preparation of DNA synthesis. So here, during the increase of the cell mass, that is the phase which has been described to be as the G1 phase, which is the quite longer phase, that is of the 10 hours, is going to be approximately 10 hours. It is going to be dedicated for the cell growth and also the chemical preparations, that is the required protein factors which are useful to synthesize the DNA. That is the semi-conservative application of the DNA is going to happen in the next phase after the G1 stage or the G1 phase. Further, the preparation is going to happen. The next slide. At the end of the G1 phase, the S phase commences, and it is of about 9 hours. The approximate time for the S phase is of the 9 hours. In certain other approximate time has been recorded to be as 9 hours. During this phase, the DNA duplication takes place. That is, the semi-conservative method of DNA replication will happen, where the one parental strand will be preserved, and according to that parental strand, the other parental strand is going to be synthesized on the opposite direction. As the DNA replication complete, the cell enters the growth phase. Again, after as the DNA replication ends, the cell enters the growth phase. That is the second growth phase is called G2 phase. This phase requires about 4 hours till the M phase, it is the mitosis. So here during this G2 phase, the overall the components are going to be going to get into the growth, and further in the M phase, the particular this molecule, the particular mitosis is going to takes place. That is the equational division of the sister chromatids are going to takes place. During the mitosis phase of equational division, the sister chromatids of each chromosome separate, and each sister chromatids gets to each of the two daughter cells, hence giving rise to the two progeny cells. hence this one you people know very well. Here the equational division of the genetic material is going to take place, where out of the sister chromatids, one sister chromatids moves to one progeny cell and the other sister chromatids is going to move to the other progeny cells by having the 2n number of the chromosomes in them. So here this further leads to the G2 phase. Progeny cells formed after division proceeds to grow by molecular synthesis, reaches a specific determined division size and again divides. Now here after the M phase, in the G2 phase, the progeny cells are going to form, that is the cytokinesis will take place, where the cytosol and all other organelles will reappear in the progeny cells, and the size of the progeny cells are going to proceed to grow by molecular synthesis, such that it reaches a specified determined division size and again divides. That is the mitosis cell cycle is going to takes place. The cell cycle is defined as the entire sequence of events from the end of one nuclear division to the beginning of the next nuclear division. Here it is such that the mitosis is such that the process of the division of the cell is considered from one nuclear division, that is the one DNA synthesis to the next DNA synthesis. That is, after one DNA synthesis, the progeny cells will form till to the next DNA synthesis, that is going to give rise to the new progeny cells. So now we are going to discuss the regulation of the mitotic cell cycle in eukaryotes, certainly depending upon whatever the slides we have already discussed. In this, still do not know the molecular mechanism by which cells measures the cell mass and initiates the cell division precisely at the right time. Still we do not know the molecular mechanism by which cell measures the cell mass and initiates the cell division precisely at the right time. And still we doesn't know how is the new progeny cell, when it has grown large enough such that it should divide and produce progeny cells. It is also not known that how a progeny cells will come to know that it has grown large enough so that further it has to take part in the cell division and to produce a progeny cells. But the chromosome's replication is very precisely controlled irrespective of growth conditions. Now here we have seen that the chromosome replication is very precisely controlled irrespective of the growth conditions. In the sense, let a cell accomplish or grow to the whatever the maximum size it has to be grown, or a little lesser, but at a particular precise time or a particular precise molecular function, the replication of the chromosomes, that is the DNA replication, is very precisely controlled irrespective of any growth conditions, and further that S phase is going to be controlled and the replication of the DNA is going to takes place. Eukaryotic cells control of the cell division is more complex compared to the prokaryotic, because it's not only the chromosome replication. As in eukaryotes, it is not only the chromosome replication, it is also about the G1 phase and the G2 phase, where the G1 phase is responsible for the S phase, where the molecular mechanisms or the changes or the preparations are going to happen, but we still don't know what molecular mechanisms and the preparations and what the differences is going to occur in the G1 phase, where which lead to the precisely accurate synthesis of the DNA, where irrespective of any growth conditions. So here this is what the difference which we are going to see in the eukaryotes compared to the prokaryotes. And also cytokinesis deregulated, but In addition, the components of mitosis should form and function at the proper time in the cell. So cytokinesis deregulation or the regulation is also seen in the eukaryotes compared to the prokaryotes, where it is the complex function, and the two at a proper time in the cell. Two key processes must take place in an accurate and coordinated manner. One is the genetic material must be duplicated by semi-conservative method. Second is the duplicated genetic material must be faithfully distributed to both the progeny cells. These are the two key processes that has to take place in the S phase, uh, S phase and the M phase, that is the after G2 phase. Further, there should be some mechanism that is going to regulate the eukaryotic cell division. How does a cell know when to initiate the replication of DNA and the chromosome condensation? Now here it's a question that how does a cell knows when to initiate the replication of DNA and the chromosome condensation? How does a cell know that the replication of DNA should be before condensation of chromosomes? It is again a question, that is how does a cell know that the DNA replication should be before condensation of the chromosomes? So there should be some mechanism of signaling that should lead to the this differentiation in the phase of the replication of the DNA at the former side compared to the later side, the condensation of the chromosomes. Studies of cell division cycle, that is the CDC, mutants of the yeast Saccharomyces pombe and Saccharomyces cerevisiae, and even in the toad Xenophus levis. In the during early embryonic cleavage divisions of these particular model organisms have shown that there are 2 points at which a cell makes a commitment to proceed through the 2 stages of the cell cycle. Near the end of the G1 phase, the first point occurs called as the start. That is, as the G1 phase ends, starts to end, and that is the late G1 phase. And at the commencement of the S phase, there is a point where it is called to be as the start point. At this point, cell commits to the initiation of the DNA synthesis. A short time later, at the onset of the S phase of the cell cycle. So, as the initiation goes on, the in the S phase of the cell cycle, the particular synthesis of the DNA is going to take place. At the very beginning of the M phase, the second point is the commitment to proceed through the chromosome condensation and chromatic separation events of mitosis. So here the at the very beginning of the M phase, that is the at the late G2 phase, G2 phase, and at the commencement of the M phase, the second point is where the condensation, the commitment to proceed through the chromosome condensation is going to takes place, and chromatic suppression events of mitosis also be going to takes place. Evidences indicates that the certain key regulatory proteins function has signals in both commitment steps of cell cycle. In Xenophus, it was discovered mitosis promoting factors are there, that is the MPF. When it was injected into oocytes, it stimulated the oocyte to enter the M phase. So here, MPF shown to contain two essential components, that is cyclins are the proteins that undergo cycles of synthesis and accumulation during G1 and G2 phase and degradation during M phase. G1 G2 and degradation during M phase. A start and M phase specific protein kinase called PP34 is the product of the CDC2 gene of a Saccharomyces pombe and the CDC28 of Saccharomyces cerevisiae. Here there are two essential components which are going to help the stimulation of the oocytes to enter the M phase. That is, one is cyclins, or the proteins that undergo PP34 protein kinase. PP stands for phosphoprotein, a protein that may have phosphate groups on the side chains of specific amino acids, and 34 stands for 34,000 molecular weight of this protein. PP34 protein kinase interacts with two cyclins, that is M cyclins involved in the commitment and G1 cyclins involved in the start decisions. When PP34 interacts with the appropriate cyclins, results have shown that phosphorylation or dephosphorylation of a single tyrosine residue may directly regulate both initiation of DNA replication and the onset of M phase. So, mitotic protein factor formation is going to depend upon the phosphorylation or the dephosphorylation of the single tyrosine residue. So this will lead to the start point formation and also the M phase commencement. The cyclins and the PP34 protein kinase are the key components, and certain homologous proteins have been identified in several eukaryotes, including in humans, frogs, sea urchins, and starfish. The knowledge of the molecular mechanism that control the orderly passage of cell through the cell cycle is still incomplete. However, some part has been described very well of the DNA replication initiation and the mitotic division of the chromosomes, that is the condensation of the genetic material in the form of the chromosomal bodies and its equational division. Hello guys, now we are seeing the schematic representation of the regulation of the eukaryotic cell cycle, mitotic cell cycle. That is, we can observe all the stages G1, S, G2, M, all the phases. In this also we can see the G1 cyclin, M cyclin, and PP34, and also the protein kinases and the complexes which is forming, that is the active PP34 protein kinase and also the mitotic protein factor. Hence this is how the schematic represented. Now we will discuss certainly what is happening here. In the G1 phase we can see that the separate factors of the where the PP34 remains the inactive but phosphorylated, where it is going to get attached with the G1 cyclin to form the complex or the factor which can initiate the S phase. That is, it is going to form active PP34 protein kinase complex, and it is going to start the commitment to enter S phase. Further, after the commencement of the S phase, the degradation of the G1 cyclin will happen. During the S phase, the synthesis of the DNA will take place, and further in the G2 phase, again whatever the complex or the second point, that is the M phase point, which is going to initiate the mitotic phase, that is the condensation of the chromosome and the separation of the chromosome should happen. For that, the PP34 phosphorylated will get attached with the M cyclin, forming the mitosis promoting factor. It is going to form a complex factor, and it is going to give the commitment to the M phase. During this commitment, the M phase is going to get initiated, and the M phase will take part in the condensation of the chromosome and also the separation of the chromosome in the equational manner. That is, the whatever the ploidy number has to be separated for that particular progeny cells is going to happen according to that ploidy number. And further, the M cyclin is going to get degraded, and further inactive pp34 is going to be suspended freely within the cell. Hence, this is how the all cyclins and the protein factor, that is the inactive pp34, forms the complexes to initiate the synthesis of the DNA and to initiate the mitotic phase, where this is going to be precisely a accurate phase which we are discussing in the different number of the concepts of the cell cycle. So these are the two main points of the cell cycle where it has been identified from the Saccharomyces bombay and also from Saccharomyces cerevisiae, and also from the toad xenophos. So this is the certain revealed evidences of the mitotic cell cycle regulation of the eukaryotic cell. Thank you guys for following Big Book, and be tuned. We will come with the new topic